Welcome back, students. This is video lecture on Chapter 5 of Vander's Human Physiology. This chapter addresses two topics. The first is receptors, and then it puts the components together into signal transduction pathways. Receptors can be considered the sensory organs of a cell. As we know from previous courses, cells must respond to their environment in order to exist. If a cell fails to respond, it will die because of inaction. One way that cells communicate is through chemical messengers. Have to be detected in some manner, and that's done by specific molecules called receptors, which are present either on the surface or within these cells. Thus far, all known receptors are protein-based, and their presence is not uniform. Different cells produce different types of protein receptors. This is a very important point to understand. Cells are bathed in extracellular fluid, which contains all kinds of chemical messengers. A cell responds to a subset of those chemistries by having its own repertoire of cell receptors, which can vary depending on the needs of the cell in both time and quantity. Two very important distinctions can be made about these chemical messengers. They're either going to be water-soluble or they're going to be lipid soluble. If they're water soluble, then they cannot go through the plasma membrane of the cell because of its hydrophobic nature. Thus, evolution has developed plasma membrane receptors, which somehow transmit the signal from the outside of the cell to the inside. However, if they're lipid soluble, then they can freely pass through the phospholipid bilayer of a cell and enter any space. And in that space, there should be a protein intracellular receptor to which that molecule will bind. Protein receptors that span the membrane are known as transmembrane receptors. This is a two-dimensional pictorial of a typical transmembrane receptor. Since this is a protein polypeptide, it has an amino terminal and a carboxy terminal, and this single polypeptide string traverses the membranes 7 times in this example, and each of these cylinders is a alpha domain with hydrophobic and hydrophilic amino acids in the correct orientation. Regardless, this will form a 3-dimensional shape that is instrumental in then relaying any signals that are detected by the extracellular loops to conformational changes that take place on the intracellular loops. The existence of domains along the polypeptide is key to its functionality. Domains enable a single polypeptide to have multiple functions. In this particular example, we have an estrogen receptor, typical of nuclear receptors. It carries several important domains, the first being its ability to interact with DNA using finger-like protrusions generated by the tertiary structure of the final protein. Then we have a ligand binding domain, which binds to the estrogen molecule itself. 2 additional domains in this example are necessary to fulfill the full functionality of this receptor. One is highly encouraged to be able to define each one of these terms and how they apply with regard to cell signaling. A couple are highlighted because of their importance to this chapter. Affinity is simply the strength with which the binding, the chemical binding between the messenger and the receptor occurs. The higher the affinity, the stronger the effect. Chemical competition for the binding site on the surface of the receptor may take place. Chemicals with similar binding properties may compete for that binding site, and in some cases the binding may be permanent. In most cases the binding is temporary because it involves non-covalent forces. An antagonist is a chemical ligand which binds to the active site on the protein, and it blocks the action, the true action of that receptor, therefore hindering any signal transmittance. Agonists, by contrast, are ligands which do bind to the active site and do initiate cellular responses similar, if not identical, to the original ligand. Up and down regulation refer to the numbers of these receptors available to be exposed to the ligand. In downregulation, the cell reduces the number of receptors. In upregulation, the cell increases the number of receptors. utilizing various means. We briefly mentioned cell specificity. Figure 5.2 illustrates that in a graphical form. We have a cell that produces a chemical messenger. The chemical messenger then migrates and binds to different cells according to the presence of different types of receptors on those cells. So in this case, only cell A has the appropriate receptor that binds the ligand, the chemical messenger, with enough affinity to induce a response. Should circumstances require, cell B could manufacture this receptor, and then it too will respond, depending on the number of receptors and their binding affinity to the same signal. Please remember that these receptors do not work in isolation. They have a very complex internal mechanism for relaying the signal from one location to another. Thus, a cell may not respond in exactly the same way if it has the same receptor to the same chemical. It all depends on circumstances pertaining to other biochemistry within the cell at that time. Figure 5.3 is a compound graph illustrating a few parameters. The dark green line and the orange line pertain to the same receptor. In the first instance, in the top curve, just the ligand and the receptor are recorded. And we can see that there's a high degree of affinity between the receptor and its ligand. In the presence of a competitor molecule, the amount of the real messenger ligand bound is reduced. If we then replace the receptor with a low affinity receptor, then we can see that the binding is further diminished. Depending on the concentration of the messenger, the ligand, the amount of binding can then be altered by these types of saturation kinetics. Whereas all water-soluble messengers, ligands, bind to the surface of the cell, the lipid-soluble ones can bind to receptors either located in the cytosol or in the nucleus. Regardless, the mechanism of action is very similar. The receptor has a binding site for the chemical ligand. The chemical ligand will enter the cell and then bind to its receptor, causing a conformational change in the receptor in most cases, and that will allow, in many cases, the expression of new genes, which will then alter the cell's behavior. In general, lipid-soluble messengers enforce their actions through a slower mechanism that changes the behavior of the cell. But once these changes are initiated, then the response will tend to last longer in comparison to water-soluble messengers, which in general don't involve gene regulation and therefore lead to a faster response through a kind of chemical cascade. They also represent a very broad range of receptors that we'll talk about shortly. The first class of membrane receptors are known as ligand-gated ion channels. They all have more or less the following properties in common. Firstly, they do bind to a ligand on the outside of the cell. Incidentally, these ligands are known as first messengers because they carry a message from elsewhere to this cell. The binding of the ligand causes the protein receptor to change internally, therefore opening a channel through which ions or other chemistry can pass. The chemical movements are dictated by gradients, thus passing from high concentration to low, and not necessarily always into the cell. Since the ligand binding site and the channel are part of the same protein, we call these types of cell receptors ligand-gated ion channels. In the vast majority of cases, their effect is to change the membrane potential of that cell. Figure 5.5 gives a representative example of a ligand-gated ion channel. In this particular example, the influx of ions causes a knock-on effect in the concentration of calcium, which then leads to a cellular response. Another class of receptors function as enzymes. Once they bind their ligand, their enzymatic domain is available to perform a chemical reaction. Many of the receptors belonging to this class are known as protein kinases. A kinase is a type of enzyme that phosphorylates other molecules by either providing or transferring phosphate groups from the environment. The typical residues that are phosphorylated in most cases are tyrosines. So consequently, these receptors are known as receptor tyrosine kinases. Their sequential mechanism of action is as follows. The binding of the ligand, the specific messenger to the receptor, causes a conformational change in that receptor. That activates the enzymatic portion. The enzymatic portion is normally located on the cytoplasmic side. Once activated, it causes the autophosphorylation of itself by transferring phosphates to its own tyrosine residues. The newly created phosphotyrosines have a different shape, therefore they become binding sites for other factors, other proteins that may be migrating within their vicinity. This then usually leads to a cascade of activity where other proteins bind to these proteins, which then activate other proteins, therefore signaling a change in the cell's behavior. One thing shared by all these mechanisms is that they activate the cytoplasmic domains by phosphorylation. Figure 5.5 attempts to convey this relationship. The ligand is in orange, the receptor itself is in blue. Its tyrosine kinase domain is indicated on the cytoplasmic surface. Conformational changes in the receptor due to the binding of the ligand result in autophosphorylation. Here's a phosphate group attached to the tyrosine residue. This now creates a unique docking site for this docking protein, and that initiates a cell's response. A very important exception to the protein kinases we just spoke about is an enzyme receptor called guanylyl cyclase. Instead of using a protein kinase activity to phosphorylate other entities, this particular receptor, once it binds its ligand, is triggered to interact with GTP molecules in the cytoplasm and convert them to cyclic GMP. The cyclic GMP then diffuses to various parts of the cell, activating in turn a protein kinase called cyclic GMP dependent protein kinase, because it depends on the presence of the cyclic GMP for it to function properly. And these kinases, they phosphorylate in turn particular proteins that are then used to influence the behavior of the cell. These indirect methods of activating protein kinases give the cell more versatility. And this is illustrated no better than in the cells of the retina of the eye, where this type of mechanism has been abundantly exploited by evolution. As another example, the interaction of a first messenger, a gas called nitric oxide, NO, which is present in blood vessels, it interacts by penetrating cells and binding to a guanylyl cyclase enzyme that is present in the cytoplasm and not located on the plasma membrane as in the previous. In another variation on this theme, the kinase activity can be deferred to a second molecule. So the receptor acts as a docking site for a second molecule, which is actually the enzyme that phosphorylates other entities. The role of these type of kinases will be discussed in alternative chapters. For now, we just need to learn the general mechanism of action of this system. The binding of the first messenger, the ligand to the receptor, causes a conformational change in the structure of the receptor on the cytoplasmic face that activates the Janus kinase protein component, which in turn targets proteins with phosphorylation. Once the proteins are phosphorylated, they then trigger a sequence of events which normally lead to transcriptional activation, and that then impacts the cell's outcome. The immune system appears to have taken this mechanism to communicate its effects on various cells throughout the body. Signaling by cytokines, which we'll talk about later, influences target cells mainly by this mechanism. The third class of membrane-bound receptors are called G-protein coupled receptors, or GPCRs. The receptor itself is coupled directly to a concoction of other proteins known as G proteins. The G protein normally consists of three subunits called alpha, beta, and gamma. It's the alpha subunit that in most cases is able to be active, although there are cases where the beta and the gamma are active too. The alpha subunit can alternatively bind to GTP or GDP, triphosphate versus diphosphate. This acts as an internal timer that tells the alpha subunit how long to be active for. In the normal sequence of events, the binding of the ligand to the receptor causes a conformational change to the receptor, so that's no different. But that change causes the G protein to lose affinity for the receptor. Therefore, the G protein is pushed away from the receptor, and at the same time, it begins to disassociate. Once disassociated, the alpha subunit is active for a period of time, controlled by its interaction with GDP versus GTP. The exchange of GDP for GTP is initiated by the binding of the first messenger and that first conformational change of the receptor. The GTP partner has two outcomes. First thing it does, it pushes the beta and gamma subunits away from the alpha, and secondly, it has a period of time before it gets broken down to GDP by an intrinsic capacity built into the alpha subunit. Once the GTP is broken down into GDP, the alpha subunit recombines with the gamma and the beta to reform the full G protein, which is normally inactive. The G protein then reassociates with the receptor, awaiting the next signal. During the time period that the alpha subunit is away from its two other partners, it is active. It's able to then bind to other proteins and change their behavior, and that includes ion channel behavior as well as enzymatic behavior. Once these other entities are activated, then the cascade of events ensues, changing the response of the cell. The G protein acts like a timed switch. Once activated, it will be on for a period of time before deactivating itself and restoring its condition back to the receptor. If the receptor happens to be still activated by a ligand, then this G protein will bounce away and continue to be active for a similar period of time before returning again and again. Once the ligand has been deactivated on the receptor, the protein and the receptor will have a more stable interaction. The behavior of these G proteins has been studied in many systems, and in most they have a stimulatory effect on some function of the cell. But in some cases, these G proteins can have an inhibitory effect as well. Believe it or not, the GPCR system, the G protein coupled receptor system, is the most numerous type of receptor family available to cells to communicate. A great example of the GPCR system is offered by adenyl cyclase and cyclic AMP. When the receptor binds its ligand, and in this example we'll use epinephrine, the result is a dissociation of the G protein, the target of which is a plasma membrane enzyme called adenyl cyclase. The cyclase then binds to ATP and converts it into a cyclic molecule called cyclic 3,5-adenosine monophosphate. And this is a very, very important second messenger that travels within the cell. Since the binding of epinephrine causes a positive effect on the cell, we then call the associated G protein a GS, and that stands for stimulatory. And you'll understand later that there are alternative ways in which the G protein can act. Therefore, this label is appropriate in this case. This particular interaction is illustrated in figure 5.6. The adenyl cyclase is here in purple, and we can see it produces cyclic AMP, which then has a multitude of effects on the cell. The levels of the second messenger need to be regulated in order to prevent a continued stimulation of the cell's pathways. A second enzyme, known as enzyme cyclic AMP phosphodiesterase, is present inside the cytosol, and that quickly converts cyclic AMP to AMP, therefore terminating the cellular response. The concentration of cyclic AMP present in the cytosol is either going to be altered by its rate of synthesis or its rate of breakdown. So the balance between the two enzymes, the adenylyl cyclase and the cyclic AMP phosphodiesterase, is important. We now know that caffeine and theophylline, the active ingredients of coffee and tea, they work as stimulators because they can partially inhibit the cyclic AMP phosphodiesterase, thereby prolonging the actions of cyclic AMP, the second messenger, within those cells. Applying terminology from Chapter 1, the pool of cyclic AMP is influenced by its production and its decay, and that pool is very important in then continuing to influence the behavior of the cell. The set point for the concentration of cyclic AMP can then be influenced by various cellular conditions. The situation is a little more complex than this because inside the cell, the cyclic AMP can bind and activate other enzymes, one of which is known as cyclic AMP-dependent protein kinase. Being a protein kinase, this enzyme will then add phosphate groups to its target molecules. Incidentally, this enzyme has an alternative name. It's called protein kinase A. Once it itself is activated, it can then continue to phosphorylate other downstream proteins. The example given here both illustrates how this system works and also reiterates that different cells can respond in different ways to the same signal. So epinephrine, a hormone, when that interacts with its receptor, it triggers the cyclic AMP pathway, which acts in adipose cells to stimulate the breakdown of triglycerides through a cell-unique enzyme only present in adipose cells. With liver cells, epinephrine also triggers the cyclic AMP pathway, but due to the fact that the target molecules are different in liver cells compared to adipose tissue, the effect of cyclic AMP in liver is to both initiate the biochemical pathways leading to glycogen lysis and also the pathways leading to gluconeogenesis, both of which are necessary to increase the blood sugar concentration. We alluded to the fact that not all G proteins are going to stimulate a biochemical pathway. Indeed, there are examples of G proteins that don't stimulate adenyl cyclase. In fact, they inhibit adenyl cyclase. These G proteins are labeled GI, and the I stands for inhibitory. Any cellular pathway that leads to the inhibition of adenyl cyclase will decrease the pool of the concentration of cyclic AMP inside the cell, and therefore the phosphorylation of key proteins. Some signaling pathways result in the magnification of the signal, a process known as signal amplification, which happens as the information is relayed from one set of factors to another. Figure 5.8 shows a signal being amplified by a single receptor a millionfold. The overall outcome on a particular cell depends on many factors. But one thing for sure, many of these components are common in these pathways. So depending on other conditions in the cell, a simple similar pathway can lead to different results. We alluded to the fact earlier that G proteins can interact with ion channels. When it comes to this system, we've discovered two mechanisms of action, either by direct regulation or by indirect regulation. In the former case, the G protein, the alpha subunit mainly, will interact directly with the protein channel without any second messenger involvement. In the indirect method, second messengers are activated by the actions of the G protein, and they then open ion channels. And the example of that is pKa phosphorylating a plasma membrane ion channel, causing it to open. A very important system for us to learn happens to be involving phospholipase C, diacylglycerol, and inositol triphosphate. In this particular system, the G protein is named GQ by the scientist who discovered it. The binding of a ligand to the receptor activates the G protein, the GQ. This then activates a plasma membrane effector enzyme called phospholipase C, PLC. The PLC enzyme further catalyzes the breakdown of plasma membrane phospholipids, a particular class of phospholipid known as phosphatidylinositol bisphosphate, and that's abbreviated to PIP2. The PIP2 is cut into two half molecules. The first is called diacylglycerol, or DAG, and the second half of the molecule is called inositol triphosphate, IP3. Both half molecules, the DAG and the IP3, are second messengers, but they work in different ways. DAG activates members of a family of related protein kinases, which are known as protein kinase C, which then phosphorylate a large number of other proteins that lead to a particular response within the cell. The IP3 travels through the cytosol and binds to receptors present on the endoplasmic reticulum. There, this molecule acts as a ligand for ligand-gated calcium channels, and these channels open in response to the IP3, massively increasing the cytosolic concentration of calcium. Calcium, being another type of cellular messenger molecule, then activates many other cellular responses depending on the conditions within that particular cell. One action of calcium ions is to activate some forms of the protein kinase C, therefore assisting the DAG. This effect is illustrated right here in figure 5.10. That then subsequently leads to the cell's response. Calcium itself is a very important molecule inside cells. Its concentration in the cytosol is normally kept extremely low. In response to cellular signals, its concentration can be elevated. Calcium is actively transported back into the endoplasmic reticulum. Thus, the pool of calcium within the cytosol is another commodity that can be used to influence cell responses. Table 5.3 illustrates the importance of calcium as a second messenger, both in a common mechanism format and as a major mechanism format. The charge on the calcium ion is very important, as that charge can influence the electrochemical potential of a cell. The calcium ion channels in the plasma membrane are opened directly in some cases by electrical stimulus, and those are called voltage-gated calcium ion channels. Indeed, the amount of extracellular calcium entering the cell by these channels can, in turn, bind to calcium-sensitive channels on the endoplasmic reticulum and cause them to open, and the combined effect is calcium enters both from the extracellular environment and from the sequestered endoplasmic concentration. This is exactly what we see in the case of muscle activation. Calcium can also chemically bind to other proteins. One common one is called calmodulin. The binding of calcium to calmodulin changes the shape of calmodulin, and then that combination can either activate or inhibit a large number of other enzymes and proteins, many of which happen to be protein kinases. The cascade of kinase activity or phosphorylation leads to the cell's ultimate response. Figure 5.11 illustrates one calcium-calmodulin interaction pathway, with calmodulin being present here. Table 5.4 lists the important second messengers that we've already discovered in this chapter, their source, and their effects. Finally, we'll spend some time looking at the actions of eicosanoids, which are a family of molecules produced from the polyunsaturated fatty acid commonly present in the plasma membrane, known as arachidonic acid. In the holistic view of things, this further illustrates the wonder of nature, where not only is the plasma membrane used for transport and for demarcating the boundary between life and non-life, also supplies molecules involved in cell signaling. These eicosanoids include the cyclic endoperoxides, as well as the much talked about prostaglandins, the thromboxanes, and the leukotrienes. Each of these intracellular signal molecules has a powerful beginning. It starts with the stimulus through a ligand of the receptor, and that receptor activates an enzyme called phospholipase A2. And that enzyme is located in the plasma membrane close to arachidonic acid. Once the arachidonic acid is liberated from the membrane, it is then free to travel into the cytosol, where it enters one of two common pathways. The first pathway is initiated by the action of COX enzymes, cyclooxygenase, then traveling through extremely complicated biochemical pathways, leading ultimately to the formation of the cyclic endoperoxides, the prostaglandins, and the thromboxanes, whilst the other pathway leads to the formation of leukotrienes through the action of an enzyme known as lipoxygenase. The family of eicosanoids is so complex that their designations are superscripted and subscripted. The generated eicosanoids may act as intracellular messengers, but for the most part they are released into the environment to act locally and immediately. These factors exert a huge influence on the physiology around them, especially with respect to inflammation. Inflammation is both a Dr. Jekyll and Mr. Hyde. It has a good side and a bad side. Inflammation leads to the battling of infections, but at the same time it can lead to tissue damage and cancer. Battling and blocking the effects of these chemicals has sprouted the pharmaceutical industry. Drugs such as aspirin, which are known as inhibitors of COX, they block the synthesis of these endoperoxides, the prostaglandins, and the thromboxanes. These drugs are properly known as non-steroidal anti-inflammatory drugs, or NSAIDs. Many times cells may wish to cease certain signal transduction pathways, and the mechanisms available are many, some of which are mentioned here. Generally, their effects are tied to the presence of the first messenger. Removal of the first messenger is one action that can neutralize a signal transduction pathway. These ligands can be metabolized by enzymes in their vicinity, or even taken up by cells and destroyed, or they may simply diffuse away after a period of time. Once the first messenger is neutralized, then internal second messenger systems can also be decreased in amplitude by similar activity, but this time inside the cell. Alternatively, instead of removing the messenger, we can modify the receptor itself. There are at least three methods by which cells are known to modify their receptor behavior. The first is that the receptor can become chemically altered, for instance by phosphorylation, and that influences its affinity for the messenger. So the messenger in this case may not bind, or once it's bound, it may be released readily. The second method is phosphorylation of the receptor in a way that prevents further interaction with proteins downstream. So G-protein interaction is what we have in mind in this case. And finally, the membrane receptors may be removed from the membrane by endocytosis. The final slide cautions us to think of a holistic system rather than an isolated system. These signal pathways are pretty complex, and we're still trying to decipher them. Their interplay is huge, depending on minute changes in the chemistry of the environment or the cell. The results can be profoundly different. The different receptors and the signaling pathways offer something known as a crosstalk, and that is very important in regulating the final outcome. Thank you so much.